Okay. Hi, my name is Maia Kinnebrew, and I am a College of Creative Studies biology major, and I work with Sonia Hussein, who's a grad student in the Department of Chemical Engineering. And my project is called Ligamentization and its effect on the function of 7-transmembrane and the 7-transmembrane protein proteorhodopsin. So what is proteorhodopsin? Proteorhodopsin, or PR for short, is a transmembrane protein that was recently discovered in 2000. So what transmembrane means is that the protein crosses the cell from inside to outside, crosses the cell membrane, and there's a lot of membrane proteins in our bodies that will cross the membrane and form a channel in the middle to basically help get molecules and ions from outside your cell to inside your cell. And proteorhodopsin specifically is found in 80% of marine bacteria, so it's found in bacteria, and it's activated by sunlight to pump a proton through its channel. So here's a little schematic. Sunlight hits the cell, which has the protein in its outer membrane, and it's activated to pump a proton from inside the cell to outside of the cell, and this ultimately creates energy for the bacteria. One interesting aspect of PR is that it forms oligomers under various environments. So an oligomer is basically an assemblage of membrane proteins that basically group together like this to perform some different function than the singular protein would. So there's a lot of different ideas. One idea is that it may amplify the signal made by the membrane protein. So why are we interested in proteorhodopsin specifically? One reason is that proteorhodopsin has a structural homology to a large class of proteins in our bodies called G protein-coupled receptors, and these proteins are incredibly important. This is the number of human genes in our body, and 57% of those genes actually code for G protein-coupled receptors, these proteins in our bodies. And like I mentioned before, they're targeted by 40% of pharmaceutical drugs on the market as well. So they're very important, but we don't know very much about them. This is because they're challenging to make, they're hard to express, and when you do express them, you don't get a lot. And they're not very structurally stable. It's hard to mimic the environment of a specific cell in a mammal. So what we are trying to do is use proteorhodopsin to study G protein-coupled receptors. So proteorhodopsin is a good model for a few reasons. First, it has the same 7-transmembrane structure as the G protein-coupled receptor. So here you can see they both cross the membrane 7 times. They also have movements on the same loop when they're activated. And they both form oligomers, so they both form these complexes in the membrane. And also, proteorhodopsin is easy to express, we can get a lot of it, and it's structurally stable. So we're hoping to study proteorhodopsin to determine some of the fundamental questions behind G protein-coupled receptors. So making proteorhodopsin takes about a week, and then at the end of the process we end up with a sample of about 30 milligrams of protein, and it's this pink protein. And in this sample we have a mix of all different sized proteins. Like I said, it forms these complexes, these assemblies, and we've shown that it forms specifically hexamers, dimers, and monomers. And so the next big important part of our purification process is actually separating these by size. So we have a fast protein liquid chromatography column, and the biggest proteins actually come out first. So we can basically separate the big protein from the small protein and then characterize them separately. So an example of work that was done previously, this work that I helped do about 1.5 years with my last mentor, Catherine Stone. We basically isolated hexamer fractions of the protein, and then we can find the interprotein distances. So proteorhodopsin is about 25 angstroms across right here. And this is actually using the technique that Mary Lou was describing earlier to measure protein distances. So this is one reason why we might want to separate a hexamer from a monomer. So this was proving to work really well, and we wanted to do more tests and see how is the hexamer different in other respects. So one thing we can do is we can activate this protein with light. Like I mentioned, it's activated by light and it's pink, and what actually happens is when we activate it, what makes it pink, this molecule right here, turns orange, and we can measure how long it takes the protein then to turn back from orange to its original pink color. So we can do this with a UV vis, time resolved UV vis basically. And so we flash the protein with a laser right here, and then we're watching how long it takes to turn back to pink. And so what we can see from this graph is basically the monomer takes about 1 second to go back to its original color, and the hexamer, which is this red curve, takes like 5 or 6 seconds. And on average, we showed the monomeric PR is about 5 times faster than the hexamer. So this is important because this is showing that the proteins are very, very different when they're in these different states and they're acting very differently. So it is important to separate the proteins. And so we wanted to go forward and do a functional measurement. And so one way we can do a functional measurement is with a pKa measurement. So we can do this because, like I mentioned, proteorhodopsin pumps a proton. And so by measuring the protonation states in this proton channel, we can determine, we can elucidate some information about its function. So proteorhodopsin actually pumps a proton right through this channel. And so we can find the pKa of D97, which is the primary proton-accepting amino acid right here. And the pKa will tell us the pH where half of the population of that amino acid is protonated. And then at pHs below that, it'll be, this is on a logarithmic scale, so it'll be increasingly more and more protonated, so here around 90% and 100%. And then at higher pHs, we're going to have more unprotonated. So like I said, it pumps a proton, and this is the active form of PR. So this residue does not have a proton bound to it, so it's ready to accept another proton to pass through the channel. And this is the inactive form. So you can see actually the proton from this retinal molecule right here hopped onto this amino acid, so it went from here to here, and now the protein is inactive and can't pass another proton. So we wanted to do a pKa experiment and basically determine at what pH is this amino acid 50% protonated. And we're interested in this because literature has already found that the pKa of this amino acid ranges from 7.1 to 8.5, and the pH of seawater, where this protein is found, is the same pH range. So, like I pointed out, this basically suggests that the protein in the pH where it lives is only half active. At this pH, half of the population has a proton bound and half doesn't. So we separated the monomer and the hexamer fractions, and we were able to find consistent pKa values for the 2 different proteins. And like I mentioned, this is a logarithmic scale. So if we have a pH of 7 where it's living, or 7.5, as we get closer, or basically this is far enough away from the pH where it's found that it's about 90% active. So the hexamer is much more efficient at pumping a proton. And the last kind of test we wanted to do, and some of the future experiments I'll be doing, is basically when we're extracting the protein from the cell membrane to study the protein, we use different environments. So these are 3 different detergent molecules. This is DDM, and this one's DIC7PC, and the last one. So a lot of different people in literature will use different molecules to separate their protein, and then they get all sorts of different results for their protein. And we are able to show that this is because whenever you make your protein in DDM, you get mostly hexamer population. And whenever you make your protein in this other detergent, you get mostly monomer. And like I showed, then you get different time scales, they have different dynamics and different functions. And so what we're going to go ahead and do next is basically combine some of these parts of the detergents, and basically we hope to determine, is it this one fatty acid chain that is making the DDM create a mostly hexamer environment, or is it more a function of the Zwitter ionic head group? So that is what I hope to do in the future.